Right now, I don't hear anything.
Okay, welcome to the section of biosensor. I would be a chair of this section. I am Bahara Azimi, and I think we can start the section with our first speaker, Frederick Cook from Lund University, with the title of Highly Sensitive Optical Detection of Biomarkers Using Light Guiding Nanowires. Frederick, please.
Thank you very much for the invitation. And it is correct, actually, that I'm associated with Lund and Nanolund. I have my permanent position at Chalmers University of Technology, located 2 hours drive north from Lund. So this is a presentation of recent work that we have done in collaboration with Lund. It is me and Heine Linke, which is focused on the use of light guiding nanowires for biosensor purposes. And the reason that I became engaged in this collaboration stems from my previous work on label-free surface analytical tools. This morning we listened to a very interesting presentation on imaging SBR. I mostly used conventional surface plasma resonance to probe the interaction of the interaction between biomolecules using one of which being immobilized on the sensor surface. My prime contribution to this field, though, is the development of the QCM device, which was also commercialized in the mid-90s. And both these techniques are now available as research instruments and much applied to address questions related to biointerface field in general and biosensor applications in particular. And after the development of the QCM technique, we became focused on the use of lipid-based surface modifications. And this lipid-based surface modification can be made either using synthetic or purified lipids. There are multiple ways by which you can make lipid vesicles and make them and use them to modify the surfaces for sensor applications. You may also use, and this has become more popular in later years, native cell membranes, which are produced from live cells and thereby even better mimic the properties of cellular membranes. The key difference between these two systems is that while one is simple and you can add components one after the other to represent the cell, the function of cellular membranes are closer to the native state, but they are much more complex, and sometimes it is difficult to deduce accurate information from the latter. So a combination of the two is good to have at hand when you develop surface-based sensors that are intended to mimic cellular membranes, and that is the expertise of my group. And I will just illustrate one example, and that is the formation of supported lipid bilayer made from synthetic lipid vesicles, which we explored a lot with the QCM technique. That is shown here in the upper left of my presentation, and this is a cartoon illustrating this process. And I won't go into the detail of what the QCM and SBR curves show today. I will rather illustrate the formation of a supported lipid bilayer using total internal reflection fluorescence, which excite fluorophores near the surface within 100 nanometer. And this is a movie of individual lipid vesicles landing onto the surface here to the left. And if you look in this region, you will see that when the density of lipid vesicles reaches a certain threshold, they start to fuse with each other and form a planar lipid bilayer. And we can even follow the rate of the expansion of these seeds to the bilayer formation. And this serves as a very good model of cellular membranes. And 10 or 15 years ago, I had my first professor position at Lund. And since then, a center of excellence has been established in Lund, which is called Nanolund. And one of the missions of Nanolund is to use nanotechnology for personalized medicine and also to contribute to the Swedish industry. And that is what I will present a progress report on the collaboration we have had with them. So today we will focus on single molecule detection of membrane-bound biomolecules using light guiding nanowires, and this is work that is related also to a spin-off from Lund called Aligned Bio, which you'll find information about on the web if you're interested. And the nanowires, this is a teaser for the nanowire system we are working with. They are gallium phosphide nanowires that can be grown epitaxially in various ways from surfaces. And to the right here, we have an illustration of fluorescent nanowires. And these are made fluorescent by binding a fluorophores to their surface. And it turned out when they started to work on these fluorescently modified nanowires in Heine Linke's group, that they had quite peculiar optical behavior. And that relates to the nature by which, under certain conditions, certain fluorophores for certain properties of the nanowires, the light can be coupled into the nanowires, which act as light guides, emitting the light at the tip of the wire. And this actually leads to an enhancement of the fluorescence, which is highly interesting from a biosensor perspective, because this allows a convenient means to increase the signal while not increasing the noise, so that the signal to noise can increase quite dramatically, and that is what we have explored in the context of bioanalytical sensing. Since my expertise area is supported lipid membranes, we started off by modifying these nanowires with lipid membranes, and the lipid membranes has a two-dimensional diffusivity, and it turns out that you can learn a lot about the diffusivity by probing how the emission pattern varies when molecules are diffusing on and off the surface. So that is the first part I will present, and then we will see whether the high sensitivity of these light guiding nanowires enables detection not of single molecules only, but also single fluorophores, and what that might bring us in the future. So these nanowires are grown by vapor phase epitaxy using gold particles as a seed, and it is the size of the gold particles and the duration of the growth that dictates or can be used to control both the length and the diameter of the nanowires. And these properties are crucial for the light guiding properties of the nanowires. And also the surface chemistry is crucial, and what is conventionally used to control the outer layer of the nanowires is atomic layer deposition, and we work primarily with silica as a coating. Any other material also possible. And they share certain similarities with optical fibers, but primarily the core here. And no cladding is used, they are naked. And the reason that they can act as a guide of light is the fact that their dimension is lower than the wavelength of light, which leads to an incoupling of light. And the refractive index, given by the fact that the refractive index of the semiconductor nanowires, gallium phosphide in particular, is significantly higher than that of the surrounding medium water, 3.4 compared to 1.3. This incoupling is illustrated in this cartoon here. And that is in particular what is used to detect fluorophores at the surface of the nanowires. And I will now show a set of confocal microscopy images of nanowires modified homogeneously with fluorophores. And with the confocal microscopy, the fluorescence emission has been probed as a function of lengths of the nanowire, or at different positions along the nanowire. This is a top view, and then the optical slicing of this image shows the following: that for a nanowire with a diameter of 130 nanometer, see that the fluorescence emission, despite the fact that the fluorophores are everywhere on the nanowire, the emission occurs at the tip of the nanowire, while if the diameter is below 100 nanometer, the emission is homogeneously distributed along the wire. And this is a feature that we thought could be used for sensor application, since the emission is also directional, which leads to more fluorophores reaching the detector. This is illustrated to the left with the cartoon. So the emitted light is coupled into the wire and is guided towards the tip where it is emitted in a directional manner. And there is also a fluorescence enhancement owing to the evanescent field surrounding these wires. But it is not automatically so for any fluorophore or any nanowire. So each fluorophore needs to be optimized with respect to dimension of the wire. And this is illustrated here for 3 different fluorophores with different excitation and emission wavelengths. And this is an example of an optimal diameter 130 nanometer for Alexa 488, where you see a clear emission near the tip, while for many other dyes or diameters you see a more homogeneous distribution. This is all presented in a paper from Linke's group. And this is an example of forming a supported lipid bilayer on a silica-coated nanowires. Now we will look at essentially the same type of movie that I showed in the beginning, but what lights up now is the nanowires. And what you see here so far are lipid vesicles that have landed as individual fluorescent objects on the surface. And as the coverage of these vesicles increases, you see that there is a collapse into a lipid bilayer, and what lights up now are only the nanowires coated with a fluorescent lipid bilayer. And what we discussed together with Heiner was whether we could functionalize this lipid bilayer with a receptor that is known to bind specifically to a protein. And we selected the receptor for cholera-toxin, GM1, and bound the fluorescently labeled cholera-toxin molecules directly to the GM1 molecules on the membrane. And what we then observed at really low coverages was blinking nanowires. It is not that easy to see on this in this movie, but if we look at it again, you will actually see that some of these wires do blink. Overlaid on that is a bleaching process, because the enhanced fluorescence leads to quite rapid bleaching. And making an analysis of this, we saw these blinking events of individual wires that they could be clearly detected. There was also a correlation between the blinking events and the nanowire position. This is a bright field image of the corresponding substrate. And by analyzing the blinking events as a function of time and excluding those wires that were light from the beginning with those that lit up after first injection of cholera-toxin, we could, from the blinking pattern, actually deduce the diffusion constant of the molecules moving in and out. We had, though, to take the bleaching into consideration. So in one model, we had rapidly bleaching molecules. This is represented by this part of the curve, while the onset is determined by the diffusion constant. And we verified this with the same molecule modified with a dye that was less prone to bleaching. And using these two models, these two regimes, we could determine the diffusion constant from single molecule blinking events, and we came to exactly the same diffusion constant, irrespective of which type of dye we used. So this was a starting, a first step towards the use of these nanowires to detect single molecules. And this is illustrated here. But each molecule here had multiple dyes, up to 5, maybe even 10. So we asked ourselves whether the sensitivity of these wires was sufficient to detect single dye molecules with conventional microscopes, because this is a normal upright microscope. This is not a laser illumination. This is normal white light illumination. So we replaced the cholera-toxin with multiple dye for DNA, with each DNA carrying a single dye molecule. And instead of having the cholera-toxin moving on and off the nanowires, we anchor the DNA with cholesterol, and then we run a movie where we detected the fluorescence emission from the nanowires. You see that to the right here. And then we bleached a fraction of the nanowires, and then we could see how they gradually started to appear bright again. And we also saw a few blinking events. So what we did was to further reduce the concentration of cholesterol DNA when we immobilized them. And if you compare these two movies now, you will see that at high concentration, they all light up, but at low concentration, there are in fact blinking events. And overlaid on that, some bleaching, but not too much in this particular case. And this blinking is a strong evidence that single dye molecules are moving on and off the wires, and the diffusion constant that we deduce is exactly what you expect for DNA diffusing on the membrane when coupled using cholesterol. And we could actually, in the high coverage regime, count the number of dyes per wire, 3, 2, 1, 0, and so forth. And in the low coverage regime, we could count the dye molecules as digital events. And this together helped us to deduce the diffusion constant. That is one thing. But what this offers is potentially a simple mean towards DNA sequencing, because what you need there is a signal when a base of a specific type is attached to a template DNA strand, and where with each base modified with a unique color, you could tell which sequence there is on the DNA. And we believe that this could be accomplished with simpler optics than today used by Illumina and PacBio and so forth. We're also very curious in using this for ultra-sensitive diagnostics, because single molecule detection should offer improved detection limits. And this we are now exploring in a center located in Lund, funded by Vetenskapsrådet, the Swedish Research Council. We're a team of scientists doing that, and we are right now recruiting to that. And we are also supported by Align Bio. And just a teaser, the last minute, is that early work using these wires for detection of biomarkers in patient blood serum indicates a 20 times increase in signal to noise and also detection limit. And we are using here not a conventional sandwich assay, but a technique that has been developed by Umenovia, a company in Lund doing protein microarrays for the purpose of cancer biomarker detection. And I'm happy to explain the details of their sandwich assays, but the key thing is that we were in this way able to increase the signal to noise quite dramatically compared with flat surfaces, thanks to this fluorescence enhancement that the wires provide. So with that, my 17 minutes are up, and I'm happy to answer any questions if you have any. So how is the questioning taken care of? I don't hear you.
Can you hear me?
Yes, now I hear you. Yeah.
There is no question. Thank you so much for your nice and interesting presentation. So we can switch to the next presenter. Let me introduce him. John Brennan from McMaster University, with the title of Aptamer and DNA Zine-based point-of-care devices for infectious disease monitoring. Please, John.
Okay. All right. Can you see my screen now?
Yes.
Okay. I will begin.
You can start.
Okay. So today I'm going to tell you a story about some work that we started back in March of this year. And if you go back and think about what happened in March, you'll realize that was the COVID-19 outbreak. And so my talk was going to be all about C. difficile and bacterial detection and things like that, which I've talked about at past versions of this conference. But I thought today I'd give an update on where we are with actually developing a home-based saliva test for SARS-CoV-2. So I don't think I need to remind everyone about the need for better testing. Just in the last 2 days, our national newspaper has said, put out a big op-ed on saliva tests could be the key to crushing this pandemic. The New Yorker this morning asked, we basically says we can solve this coronavirus test mess now if we want to. And that was basically saying we need home-based testing and rapid, very simple and cheap testing. On the top here in the middle, you'll actually see Abbott's original assay that came out back in April and was demonstrated at the White House, which was their LAMP-based assay for doing SARS-CoV-2. This week, Abbott announced their new COVID testing card for doing home testing. I'll mention something about that in a few minutes. But sort of ideal situation is to try to develop something that looks like a pregnancy test that you can use at home using saliva as a sample. So at this point, there actually is no home-based, and that should say home-based saliva test for SARS-CoV-2. If you look at the current testing and the way it's been done since the beginning, generally you go to a central site, you get a nasopharyngeal swab jammed up to the back of your brain, and you then take that swab, put it into viral transport media, and over the next maybe 2 to 8 days, a lab will run a reverse transcription polymerase chain reaction or RT-PCR assay and eventually give you a result. The issue there, of course, is that if you do it at the wrong time, if you're too early or too late in the infection, you won't catch it. And also, this now means that if you have an 8-day period of turnaround, for those 8 days, at least in Canada, you have to self-isolate. So to address this, Abbott has just recently, in the last 2 weeks, put out the Binax Now lateral flow device, but this still requires nasal pharyngeal swabs as the sample. So this can only be done at a clinic because you cannot generate or collect your own MPS samples. Yale, a few weeks ago, talked about a saliva test. So this works with saliva samples, but you have to collect the saliva at home and then send it in, and it's tested by the standard RT-PCR method. So again, this can only be run in a centralized lab. So again, right now, there is no test anywhere that you can do at home where all the steps are done at home. So what people are looking for is the home-based saliva collection, followed by something that looks like a simple pregnancy-style test. So we started working on this in the middle of March and actually started getting funding for it in May. So for 2 months, we were doing this by bootstrapping. So the current version of the test is what I'm going to talk about today. So what we now do, we use existing saliva collection kits. So this is one shown over here. So basically, you take what looks like a sponge. This goes under your tongue, and it just has a holder here. You leave it there for 2 minutes. You take the sponge and put it into a syringe. You put the cap on the syringe, and then you squeeze the sponge out into an Eppendorf tube. So the sponge is a really useful thing because it avoids picking up other things like sputum, et cetera, that you can get if you use drool. And it also gives you a relatively consistent volume. So we then use a very simple viral lysis step, which I can't talk about today because we haven't patented it yet, but to liberate the genomic RNA. So if you remember what the virus looks like, it's just a big ball of lipid, but the RNA is inside of the lipid sac. So you need to bust open the virus in order to get at the genomic RNA. So we have a method to do that we think is compatible with home use. We then use something called a 1023 DNAzyme, and I'll talk about how this works in a few minutes, to selectively cut out a very specific piece of the genomic RNA. The liberated RNA is used as a primer for a method called rolling circle amplification, which can be done at room temperature. And the output of the RCA is run onto a lateral flow test to give a test line. And within that, we've also built all of the appropriate controls and so on. So that's the basic idea. So the question now is, does it work, and where are we currently sitting? So the first point is, can we actually generate a RNA primer for SARS-CoV-2? So the way we now do this vial 1 is this vial here now from our saliva collection. So vial 1 has within it the 1023 DNAzyme in a stabilized form. So the virus is put in here, the saliva is already in here. We run the lysis, and that liberates the viral RNA. And then the DNAzyme that's already present in the vial basically takes this, and there's an enzyme in there, PNK, that we need in order to make this work as a primer later on. We have to get rid of a cyclic phosphate off the end here. And this should then generate a very specific RNA fragment. So first thing we'll just talk about briefly is, what are these DNAzymes? Well, anything that is considered a functional nucleic acid would be considered an aptamer. This is a sort of DNA-based antibody, or a DNAzyme, so a DNA-based enzyme. So the way to think about this is, if we look at just a single strand of DNA, we can within it have self-complementary pieces, so we can get structures that start to form. So this is a stem loop or a hairpin, but we can also get much more complex structures. So this is a 4-level G quadruplex. This is a 2-level G quadruplex. And we can also get structures that fold in very specific ways that have a function where, if you look here, you'll see this is all DNA, but this little r right here is an RNA linkage in an otherwise all DNA system. And what can happen is, this piece is catalytic. It can cleave this ribo linkage. So this is shown here, and this is the actual 1023 DNAzyme. So what you see here is all DNA again with a single RNA. So this is how these were originally designed. But now imagine that this is just genomic RNA. So these are all ribonucleic acids. And what we do is we design these two pieces here and here to hybridize specifically to a very particular part of the RNA that's the genomic SARS-CoV-2 RNA. And then this catalytic piece will come in, and it'll cleave at a very specific point. What this does is it liberates this piece as a primer. And so that primer now can go on to do the RCAs. We'll see in a minute. So the first question is, does this actually work? So what we basically have here is uncleaved RNA. And this is some earlier work. This is not the full genomic RNA, but we've now done this with genomic RNA. And so you see on a gel that we have an uncleaved RNA band. When we add the DNAzyme, which is represented by these pair of scissors, it cuts the RNA at a very specific site and liberates a specific sequence that we now know can be used as the primer for this RCA reaction. And the idea here is we're targeting a GU site, in this case of the N1 site of the genomic RNA. So we know we can get the RNA primer generated. And what we then do is we take this and transfer it to a second vial. And this contains a circular DNA template, 529 DNA polymerase, and DNTPs. And what will happen is the RNA that we liberated in step 1 binds to the circle, and the 3' end gets extended by 529 DNA polymerase, and it goes over and over and over. So this also has DNA displacement or helicase activity. So when it reaches the end of the circle, it just displaces the original DNA that was synthesized and keeps going. So this then generates a very long RCA product. And this is a polymerized product that has many, many thousands of bases. What we're able to do is encode into this circle here whatever it is we want as the antisense strand to be generated. So the question now is why RCA? So first off, it's isothermal. There's no need for temperature cycling like you would need to do in a PCR. It works at room temperature. For a home test, we don't want to have a situation where someone has to heat their sample up to 65 degrees like you would with LAMP. Requires minimum number of reagents and primers, as you've seen, and it is compatible with DNA aptamer or DNAzyme biorecognition elements. So that's why we use it. So what we now know is we use these RNA cleaving DNAzymes, RCD, and this shows the cleavage site here. When the DNAzyme interacts with, in this case for us, this is an all RNA substrate, we get this cleavage to happen. We generate this primer. The primer binds to the circle with the 529 and off it goes. And there are multiple ways of doing this. I'm not going to go through the second method here, but the key here is this cleavage reaction up front generates a primer that binds to a specifically designed circle to cause this reaction. What makes RCA also really interesting is that, as I mentioned, within this circle, we can encode the antisense to whatever we want. So we can just look at using a fluorescent intercalating dye and have a fluorescence output. We could use peptide nucleic acids with labels on them. We can form G quadruplexes as the output and get things like peroxidase mimicking DNAzymes, that's shown here. Or we can bind fluorescent dyes to the G quadruplex. We can actually incorporate fluorescently tagged DNTPs that will be covalently inserted. We can use cDNA with the fluorescent tag. We can use molecular beacons. We can actually generate new RNA cleaving DNAzymes or bind gold particles. And so another thing that we can do here is we can actually build in restriction sites and monomerize this if we want. So the beauty of the RCA is we have all kinds of potential to generate different outputs. So the question we now wanted to look at was, can we use the RNA that we generated in step 1 using the 1023 DNAzyme and have it interact with the circular template and all the other reagents to generate an RCA output? And if we look through here, we see only when we have our RNA, the DNAzyme that does the cleavage, the circle, and the PNK all have to be there, and we see an RCA product. And in this case, what we did is we increased the amount of RNA, and we simply added an intercalating dye, and you see that the fluorescence goes up. And again, you can see that within about, say, 15 minutes, we can get a reasonable amount of fluorescence from this assay. So it's reasonably fast, and we have spent some time optimizing that component as well. So the next question is, how do we take this and convert it to something that's easier to see? So we can look back at all of these various outputs, but obviously anything involving fluorescence, you're going to need some kind of a handheld reader. This makes it less amenable to home use. This method here generates a color output. It goes from colorless to blue. It's basic peroxidase DNAzyme assay, but you need to add peroxide at some point in order to get this to work. So what we thought was, let's use a method that operates using gold nanoparticles, and that's generally going to be lateral flow type detection. So the idea here is we've now generated our RCA product. This is a long polymeric piece. And what we also have in our vial 2 is a toehold DNA hybridized to something we call bridging DNA. This is the piece that we're going to detect on a lateral flow test. So what will happen is this RCA product has encoded into it the sequence that is complementary to this toehold DNA. So the toehold DNA will bind more strongly to this RCA product than to the bridging DNA. What this means is the bridging DNA gets liberated, and this can bind to complementary DNA with a gold nanoparticle. This is already present on a lateral flow test. And this is one half of our sandwich. There's this extended end here, which flows up the lateral flow device and binds to a second complementary DNA that's covalently immobilized on the LFD. So when this happens, we should see a test line. So does it work? Well, the first question is, does this release of the bridging DNA by the RCAP work? So we love doing gel electrophoresis. I think we must run about 100 of these a day at this point. So this basically shows there's your toehold DNA. There's where your bridging DNA should show up. There's what we see when the two of these things are together. And if we take a monomeric version of the RCA output, we see it there, and we see our bridging DNA liberated. If we now take the actual RCA polymeric output and run it, there's the bDNA. So it's been liberated. And the proof is sort of in the pudding. So what we now see here, buffer, we don't see anything. The bDNA, tDNA, you actually see a bit of the bDNA, you see the line is what you're supposed to see. If this is left intact, you don't see a line. This is what we expect. If you add the RCA, either monomeric or polymeric output, you see the test line. So at this point, all of the pieces are working together. And the question then is, what's left to do? So what we're now in the process of doing is moving from genomic RNA, which we extract through a method that's not amenable to home testing, towards now deactivated and eventually to live virus. The live virus, the issue is we need to work in biosafety level 3. And I can tell you right now, getting access to BSL-3 labs is almost impossible because all the people making vaccines are using them. We have already evaluated many, many more DNA zymes for optimal performance. We've screened over 200 of these so far, and we have them for different parts of the gene, so the spike protein, the N protein, et cetera. We're looking to multiplex the output, so have multiple genes show up as multiple lines so that we'll be much more convinced that if we see a positive test line, it's real. We've done quite a bit of work to optimize the RCA method and increase its speed because we want to use reduced amplification time in order for this to work. And of course, we need to, and we're already setting up to do this, we need to run a comparison of patient saliva and patient MPS samples in order to validate the test. So this trial is currently being designed. One of the, I guess you can look at it as a fortunate outcome. In Canada, we don't have very much in the way of positive tests, maybe 300 or 400 a day. And so what this means is getting access to saliva samples from positive patients is actually quite challenging. So it could take a bit of time to finish this part. And so with that, I will end my talk, and I guess I have a couple of minutes for questions.
Okay, thank you. Can you hear me?
Yeah, kind of, yeah.
Okay, thank you so much for your great work and for your nice presentation. Let's see if there is any question from the hall. And let me to check the chat. So there is, I think there is no question. Okay, thank you so much.
Okay, yeah, I don't see one, but.
Thank you. So we go to the next presenter, who is Markier Graduski. I hope I pronounce in correct way. So the title is a portable quality control lab from invention to product prototype. Please, do we have?
Oh.
I think he's not connected yet. Maybe.
gestire questo piccolo lanciato?
Is not still connected.
Non included.
Okay, he's not still inside. We
Not
wait
inside.
a few minutes for him.
Che si vede piccolo. Non so. Non so. Quello sotto blu. Questo celeste. Questo. Il submit. Questo per inviare. Ok. We can try with the next presentation. I don't know. Ah, così. Così. Ecco perché. Basta così. Fai control e poi ruoti. Ok. Così ingrandisci e diminuisci secondo come ti serve a te. Infatti. Try to call him. The informer. Okay, he's not here, but
No, he's not here. We wait for him for a few minutes. Maybe he joined us in a few minutes.
yeah, we can place our Giacomo, but we will see. Sorry. Okay. C'è Giacomo?
Okay. So we have Maki.
Hello.
Okay. The hall is open for you. Please.
Hello. Can you hear me?
Yeah. We are waiting for your presentation.
Yes.
Okay, it's connected.
Okay.
Yes. Hello.
Hello?
No, wait. I'm here.
There he is.
No.
Can you
We
hear
are
me?
waiting for
Really?
must be waiting for your presentation.
Yes. Okay.
I'm going to repeat this title again.
Yes. Hello.
Portable Quality Control Lab from Invention to Product Prototypes. Can you share your screen?
Yes. I was. Okay. I think that you see now my screen. Could you confirm that you see everything? Are we in the presentation? Okay. Welcome, everybody. Sorry for this small technological hiccup. Apparently, I followed the wrong link. I would like to talk to you about Portable Quality Control Lab, and I would like to bring you from the conception of the invention to the current state of our prototype. And as many stories, this one started as well in the local bar in Groningen. This is in the northern parts of the Netherlands, while I was doing a PhD. And of course, after hours, we very often stroll down the street to a bar, and we made a couple of doodles on a beer coasters that actually this particular one turned into a patentable invention. We drew as well a couple of concepts about the company that we wanted to start after our PhDs. But as well, we are thinking in which direction this company should be going, and we're looking for an opportunity in analytical chemistry world, not necessarily in the healthcare, but definitely in life sciences. We started looking for what is done outside the academia and what molecules are being tested, how many of those molecules can be tested in professional labs, and it turns out that it's a great number, more than 200,000 different molecules. It's routinely tested in different labs. And as you can see that as well, an amount of money that is being spent on those tests is incredible. And with the current technological developments, this market is growing quite rapidly.
Sorry, stop you. We cannot see your screen now. Can you please again share your screen?
Yes, I'm doing it as we speak. Yeah, I have a communicate that I'm sharing. Yeah, do you see it now?
That's okay.
Yeah, it's okay.
Yeah.
Yeah, so we discovered this big potential in not necessarily medical market, but environmental market. And we started thinking what kind of equipment serves this market. And of course, like every market, there are two different spectra, something that you can purchase very cheap, handheld devices, maybe not the most convenient in use, but as well you can spend hundreds of thousands of euros or dollars on the equipment and trained personnel to achieve much better results. Of course, there's a niche that is being filled up with more and more advanced technologies at relatively affordable price. So that's, of course, where we want to be placed as well. But as well, we asked already different people that are involved in testing environmental samples, how they would describe those cheap devices and how they would describe those super expensive, and there are adjectives that they appeared most. So tedious work versus very reliable and precise, not necessarily user-friendly. So therefore, we started thinking about something that can be used by multiple people without extensive training, and will make those people happy in a sense of the amount of work that they have to put in it, and as well they would trust those results. And of course, what is filling up this niche in between cheap and expensive is something that we refer as parameter-centered. So that means that there's one device serving for tests for only one parameter. What we would like to do, we would like to develop a device that can run multiple tests, can accommodate multiple tests in one readout device. And as every startup, we have to be cost efficient. We need to be able to optimize different tests to a uniform format that can fit in this universal device. And we would like to move towards the cloud-based database system, and as well artificial intelligence that will will act as a trained analyst for our customers. So these three pillars, when we were thinking how to realize them, how to execute them, we got to, if you want to do something cheap, you want to use cheap materials to start with. We figured out that we can move towards paper microfluidics, or very similar or very close to paper microfluidics. We have different lateral flow assays. For the adaptation of different formats of the test and convenience of use, we are using 3D printing for rapid prototyping, and we work on our software development with software engineers. So chemical tests on paper, they are referred as easy to use and store to distribute. Paper as a very porous network full of capillaries can actually accommodate and store different chemicals safely for extended periods of time. Paper is as well chemically inert, which makes it very nice matrix for chemical deposition, and can be modified. So it can appear in the form of powder, but as well we can bend it in different forms and shapes. Just to mention, so there's more than 200 different types paper, commercially available types of paper that can be used for analytical chemistry. For 3D printing, we primarily use SLA printing. This is laser sintering method, where we use light to cure liquid resin. That gives us freedom in many iterations in our device. We can create leakage-proof structures. The resolution of those devices allows us to work at a microfluidic level. With a little of investment in the 3D printing, we can achieve really good reproducibility for our devices. However, these two parameters, cost efficiency, with this type of printing, it definitely doesn't check this box out. So we had to already, at this very early startup stage, we had to move to different fabrication methods for different elements of our product that I will show in a moment. For software, there are, again, we do everything in freeze. So this pillar has to be able to perform our tests automatically. The software will be improving with data input. So the more tests you perform, the better software you will get, and all the data has to be safely stored in a cloud available for our future customers. So as every startup, we started at our desks at home. So you can see that we are using commercially available Raspberry Pi, couple of cables, 3D printed object, just to check the concept if we can reproducibly image different chemical tests based on lateral flow concept, and how we can analyze those data. So that was the origin of our universal readout platform. And if you remember this doodle from the first slide that I showed on the coaster, this is actually patented device, which we call sample concentrator, which allows us to accumulate all the sample that we collect in a pre-selected region for the analysis. How this sample concentrator works, I think everybody spilled at least once in their life tea or coffee on a piece of paper. So you are familiar with so-called coffee stain effect. There's a drop on paper which essentially sips into a paper, and on a brim of this droplet, the signal intensifies because liquid is being pulled by capillary forces to the outer rim. So when we don't facilitate this effect, with the descending dilution, you start losing the signal of the color. In this case, it's a food dye. However, if we enhance this effect by selective drying in a pre-selected region, we can pull all the liquid that was contained in this droplet to a region which later will be captured and analyzed by imaging. So you can see that on this simple food dye, we can already improve. We improved compared to no cartridge 1,000 times. Oh, sorry. So what happened next? So this device, after patenting, we figured out that we can't keep 3D printing all those sophisticated channels for the air distribution. So therefore, we split up the paper. Paper assays are contained in cartridges or pods. Just to relate to, for example, this espresso machine that you use, everything is contained in one pod. You just plug it in the machine and the coffee comes out as a result. In our case, the chemical reaction occurs as a result, which is being orchestrated by this sample concentrator. So how it works, the pod itself, so when we relate to lateral flow assay and, for example, pregnancy test, it's very simple in use, right? You just have to put a droplet of urine and you get a signal as a result, which is very easy to assess. When we speak about more advanced chemical parameters, very often the quantitative information is needed. So therefore, this system for the quantitative analysis requires more steps to perform the chemical reaction. For example, precise dosing, elution, sample cleaning, and all this has to be somehow fulfilled, has to be encapsulated in a device that will work reliably in inexperienced hands. So we have different modules that they fulfill different functions. So assay module, I will start with the yellow one. This is the place where the chemical reaction occurs. When it's done, the signal is being displayed on the bottom, and sample is being delivered and dosed in the red module and eluted, if necessary, by a buffer or solvent contained in a blue module. And this is how it looks. So blue module is out from the picture for now. We have an open basket into which we pour a bit of a liquid. When this part is done, then we can close it with a blue module. Here you can see that this is actually the only area where we store our chemical ingredients. We can pattern surfaces with different chemicals in such a way that we can orchestrate it, the chemical reaction up to the laboratory standards. And this reaction after closing with a module, the pod is being plugged in a machine, and the machine carries out the test for you. So you don't need to be bothered anymore by any further actions necessary to perform the test. These blue arrows indicate concentrator at work. So the concentrator works in two ways. One way is to make sure that the entire collected sample is being analyzed. So all the liquid trapped here has to finally travel till the end and will be evaporated. But as well, it works as an accelerator of the reaction because we actively evaporate. So instead of waiting for everything to sip through, we accelerate the reaction by the factor of 10 at least, dependently on the packaging of the column. We verified this concept with very simple pH testing. So by deposition of phenol red or bromothymol blue on a piece of paper, we ran it in our pod system and checking it our readout device. We cross-checked it, everything with laboratory standards. So what was used for this testing? So sample was simply a buffer with a known pH. A couple of words about analytical platform. So this image we've already seen. So this is how we started. And at the moment when we purchased our first 3D printer, we started developing the prototype, which started expanding not only in size, but as well in the capabilities. So now we are able not only to take images and modify the illumination of our sample, but we have a motorized stage that ensures precise positioning of the sample. We can control the stream of gas that's necessary to operate the sample concentrator. And as well, we have a touch screen for the ease of use for our future users. So like this prototype number 2 was 85% printed. Prototype 3, it was 90% 3D printed. When prototype already 4 and 5, the entire interior is already made out of aluminum, which makes everything much more stable and reliable. Which I don't need to mention, but for the future product is very important. So there's a couple of capabilities of this platform that I mentioned, just to recap. We do as much as possible to automate the chemical reaction for our users and as well test readout. Everything is displayed on a touch screen, and the controlled gas flow for our invention at least is very important. So why all this, and actually where we would like to sell it or who could actually use it? We turned our heads into, again, food. We are beer lovers, we are coffee lovers. We are really crazy about all types of food. So we started thinking with this artisan movement around, especially beer, why not to try to prepare such an analytical platform for craft brewers? So that's our first business case that we are attempting. We already verified it with a couple of local breweries. We are running tests for them. Those values for the brewer are already checked by us on a small brewers population. And that's the direction where we are moving. The parameters that we want to test first, they are based on the brewer's feedback. So as 5, the most important parameters, actually 4: pH, alcohol content, sugar content, bitterness and different minerals in water, everything in one platform, just in different pots. And I think that with this, I would like to thank you, of course, my research group at University of Groningen, headed by Professor Verporte, and my startup team. This is the guy that I was drinking beer in the bar mostly, and our supporters, local breweries, local investment funds, and local European organizations. And my contact details if somebody would like to reach out to me after the lecture. If there are any questions, I'm happy to take them. Thank you.
So thank you so much for your nice presentation. Let's see if there is any question. So, and from the chat, there is no question. I have just one small question. As I understood, the advantage of your method for your 3D printer is fast prototyping, yes?
Yes, that's correct.
Okay. In comparison to the other 3D printers?
Yes, so we use 3D printer because at this initial stage, our device consists of hundreds of different parts. So if we are making one tiny alteration, it requires multiple alterations in different parts of the design. So therefore, if we want to move fast, working with 3D printers is the most efficient, though it's definitely not a final solution.
Mm-hmm. Okay, thank you.
Thank you very much.
So I think we can close the section and we have the break, and we will start at 16:10 for the next section. Thank you so much. Since we don't have any other
Ok. Ciao.
Hello?
Ah, ciao
Hello?
professore.
Can you hear me?
Yes, I
Eh,
hear you. You hear me?
no, it's, you are mute, now.
Yeah, just
Ok. Da, quindi fra 5 minuti si comincia.
Okay, good.
Perfetto. Perfetto.
Okay.
Start to speak.
So,
Let's go.
Yes.
good afternoon. We can start the next section, advanced analysis. So we start with our first speaker, Giacomo Ceccone, with title of XPS and TOF SIMS analysis of functionalized carbon and gold nanomaterials for bioapplications. Please, you can start.
Thank you. Can you hear me?
Yeah.
Okay, good. So thank you very much, and good afternoon, ladies and gentlemen. I'm Giacomo Ceccone. I'm working in the European Commission in the same lab of Pascal, and I would like to thank Pascal, Serena, and Ugo for organizing this. Unfortunately, I cannot be in Pisa, but that's life. So I will try to tell you something about our approach for surface analysis of nanomaterials. I will speak about functionalized carbon mainly. The gold is something that I put in the abstract, but I was a little bit too optimistic in having 17 minutes and try to talk about all these materials. So the outline is just a short introduction about nanomaterials, and then why surface analysis of nanomaterials is important. And then the application of X-ray, photoemission spectroscopy, and time-of-flight secondary ion mass spectrometry to analysis of carbon nanoonions, and some conclusions. So, well, nanotechnology in our world is definitely here also in this very bizarre time. As you can see from these 4 examples that I pick up from internet, in where the people are essentially trying to use nanotechnology to detect or to cure or to prevent the possibility of infection by the COVID-19, as you can see, for instance, in the bottom left, with the mask and the individual protection equipment, or using biosensing to test and to try to detect the virus. But in general, nanotechnology has been around us for a long time, and is employed in several products and applications in the world. For instance, the cosmetics, food, paint, and also other kind of products, and in medicine too. So the market was essentially foreseen very, very positive in the last between 2000 and 2020. They were essentially saying that you will expect an increase of $3 trillion of US dollars. But after some time, they realized that basically applying nanotechnology to some fields, and in particular medicine, is not so straightforward as it can result from the first type of study. And so there are now 2 scenarios in terms of market. One is more optimistic estimating, and the other one is a more skeptical estimation of the global market in nanotechnology. But now with the COVID, we have still, I think, to rethink about this estimation, because this kind of time will change a lot this type of way to introduce nanotechnology in our life. So, well, nanotechnology and nanomedicine, why the people were interested in using nanotechnology and nanomedicine is simply because the first thing that the people was thinking is that because the dimension of the nanomaterials and in particular the nanoparticles are very similar to the dimension of biomolecules that are moving in the cell and in the human body. So the idea was essentially to use nanomedicine to do nanoparticle to do therapeutic nanotechnology, diagnostics, in vivo imaging, and also drug delivery. So the people essentially was dreaming to have the possibility to use, for instance, what I call a PowerPoint nanoparticles, which has inside a magnetic iron, as we saw in the last talk, where the people were using this for hypothermic and magnetic resonance imaging, then surrounded by silica, for instance, a protection, because the silica will protect to eventually release of dangerous ions. And then gold, because gold is something easy to grow, and also it has very good light properties, and it can be easily functionalized with different kind of groups like polyethylene glycol to avoid agglomeration and to avoid sequestration by a certain kind of proteins, and also from biomolecule to target certain type of cells or certain type of receptors. But in fact, what you come out normally is something more like that, in which the nanoparticles are definitely not spherical. They have different kind of faceting, and the functionalization is not uniform, like you can see in the cartoon, but it's definitely something more dense in one facet, something less dense in the cuspids. And also, when you go on the right side, you can see that increasing, for instance, just the gold, you increase the faceting, and you have the tiols, which are essentially bonding to the gold in a different way, depending on the dimension and on the structure of the particles. So why nanomaterials and surface analysis are meeting, or where? Well, they are essentially meeting because most of the nanoparticles intended to use in nanomedicine or in medicine in general, even if used in medical devices, are not the bare nanoparticle, but they are functionalized with some chemistry, mostly covalently, but also electrostatically. So most of the paper already in 2008 were analyzing by this review by Dave Granger and Dave Kastner. This paper demonstrates that a lot of papers didn't report any rigorous surface analysis and characterization to validate the structural properties of biomedical application of nanoparticles. More in general, there were essentially a lot of study by people that were essentially using surface analysis for catalytic studies in the 80s, where they come out with some general notes in which the size is essentially the one properties of the nanoparticle, nanomaterial that can be very good for definition, also for legal and political issue, but is definitely not sufficient to determine the properties of the nanomaterial. And surface chemistry composition must be assessed. In this respect, I can give you this title, which is a characterization of nanoparticles, where I contribute, edited by Elsevier, published at the beginning of this year, where a lot of scientists contributed to different techniques to analyzing nanomaterials and nanoparticles, including surface analysis. The other problem when you do or you try to do surface analysis is because it's related to the provenance paradigm, as it was essentially indicated by Don Bayer from the Pacific Northwest Lab. So when you analyze nanoparticles or nanomaterials, you need to know where the nanomaterial come from, how it was stored, or how it should be stored, because the nanomaterial are dynamic. They are not static and stable elements, and they can be dynamic like chameleons, so they can change with time. And this has been already demonstrated in the time of catalytic and catalysis reaction in the 80s, but it came out very strictly for instance, when you analyze cerium oxide nanoparticle, where the cerium oxide is able to change between 3+ and 4+, even during the analysis. So it's very difficult, and it can be very challenging to analyze and to determine the chemistry of the surface of a nanoparticle. So given that, I can pass through the carbon-based nanomaterial. So this carbon-based nanomaterial, they rise up 2 Nobel Prize, one in the 96 for the fullerenes, and another one in 2010 for the Nobel of Nozelov and Game for the graphene. And there are a lot of other type of nanodiamonds, for instance, nanofiber, nanocones, nanotubes. Those kind of material are very interesting for several applications. And the people were very excited in trying to use that in energy storage and conversion, for instance, as a catalyst, or in transport because the mechanical properties, and also in as a structural material. But people also try to understand if there was a possibility to use this nanomaterial as a functional material in biomedical engineering. Amongst the different type of carbon-based nanomaterial, one of the material that demonstrate a quite good promising properties and promising structure to be used in nanomedicine is what they call the carbon nanoonions. Carbon nanoonions are basically something that has been discovered or essentially made the first time in 92 by Ugarte, where essentially he was treating some nanodiamonds at a very high temperature and then realized that he came out with some particles which were essentially circular layer of graphite and graphene with a core, a void core about 1 nanometer. Those nanoparticles were quite interesting because they were extremely nice in terms of distribution in size. They were essentially between 5 and 8 nanometers, and also they were quite easy to be produced by nanodiamonds treatment, about 95% of efficiency. So the people start thinking how can those nanoparticles be used in terms of nanomedicine and which application, other application. And as you can see, they were tested for catalysis, electromagnetic shielding, and also in optical. One of the problems we have with these particles, though, as the usual nanomaterials based on carbon, is that the poor solubility in aqueous and organic solvents. So basically, you need to treat them in order to make them soluble, and you need to functionalize them in order to make it suitable for different applications, in particular in nanomedicine. So this work I'm going to show you now is done. It was done in our laboratory, and basically with the presence of Silvia Giordani, which is now a School of Chemical Science in Dublin City University, but at that time it was at the Istituto Italiano Tecnologia in Genoa, in Italy. And with Valentina, did almost all the surface analysis, which is now at ADIMEC. She was postdoc in our lab. And 2 other collaborators of Silvia, Mario Frasconi and Riccardo Marotta, they were done basically the TEM and optical spectroscopes. The idea that came out from Silvia was essentially to double functionalize the carbon nanoonions using 2 types of functionalities. One, a fluorescein to do the imaging unit for detecting these in cells, and the other one, a folic acid, which is the receptor target that bind the folic receptor in the cell, which are essentially attacked by cancer. Most of the cancer cells essentially are quite sensitive to this kind of folic acid. So the idea was to use a click chemistry to attach the fluorescein and a kind of other quite complex reaction using essentially a carboxylic reaction and to attach the folic acid receptor and put together a polyethylene glycol spacer in order to give this molecule much more freedom to move into in respect to the core of the carbon nanoonions. So how the these materials are usually characterized. They are characterized with the usual technique: DLS, zeta potential, IFM, FTIR, Raman, because with the Raman you have the ratio between the graphite and graphene and sp3, sp2 bonds, and also TGA. So in our case, the material we receive or we have used are essentially they are treated by nanodiamonds and are 1650 for 1 hour in helium, and we have essentially this kind of nanoparticle as is shown by TM, so about 5 nanometers with 6 to 8 layers of graphite and graphene, and the inter-layer distance is about 3.3 Ångstrom. So the problem with nanomaterials is that all these techniques that I showed you before, they give you a lot of information, but very indirect information about surface characterization and surface chemistry. The idea was to use XPS and SIMS to see if we were able to characterize all the steps that were essentially involved in this quite complex functionalization of nanoparticles. So this is the system we have in our laboratory in ISPRA. So it's comprising of an XPS, our TOF SIMS on the left side, time of flight SIMS 4, and an XPS from Axis Ultra on the right side. The system is connected with a radial chamber, so you can transfer sample between the two systems without breaking the vacuum. Those techniques are working at ultra high vacuum, and so this is one of the problems when you have to prepare sample, because you have to dry essentially sample from a solution most of the time. So how the carbon nanoonions are essentially made and functionalized, okay, these are, I don't have time to go into detail, but these are the different steps to functionalize basically the carbon nanoonions in order to be ready to accept from one side the folic acid, and from the other side, sorry, from this side the folic acid, and from the right side here the fluorescein, together with the PEG chain spacer. And these are the other steps that you have to do in order to functionalize this material and to end up with something which is essentially comprising all these complex functionalities. So as you can imagine, these steps are not easy to do. You have to synthesize the molecules, attach the PEG to fluorescein and to folic acid. You have to attach the carbon nanoonions, and each step needs to have a lot of washing and redistributing the carbon functionalized nanoonions into solutions. So with the Raman and TGA analysis, we are able to check the fact that the functionalities are there. You see that there is a decrease in temperature with the TGA depending on the pristine carbon nanoonions when you go with the carboxyl functionalities, and when you go to trimethyl siloxane functionalities here. And the Raman show a different ratio between the D and G bands, which is essentially something that has been already noticed in other kind of carbon-based material. When you try to functionalize, there is a decrease in the sp2 bonds and an increase in the sp3. And with the TGA, again, we can still follow the functionalization, and essentially we can see that there is a further decrease in temperature for when you functionalize with the folic acid, and then when you end up with the fluorescing stuff. And from the TGA, you can also try to estimate the number of functionalities per carbon based on a simple model. But on the other hand, is an indirect method, because it doesn't tell you that you have the fluorescein is completely attached. It doesn't tell you if how much fluorescein you have, or respect to the folic acid, if there is a covalent bonding, if the click chemistry had worked. So you know that you have functionalized, you have put something on the surface. So for the TOF SIMS XPS, the first problem is the sample preparation. And this is a very difficult task in some cases. And also with this material has been quite tough, but we were able to use gold substrate and Teflon substrate, which are essentially were essentially quite good to form a uniform layer in order to avoid any kind of contribution from the substrate, or limiting the contribution as far as we could. So XPS give some quantitative indication about the functionalities, and in particular, when you start doing the trimethyl siloxane functionalities, you have basically an increase of silicon content, an increase of nitrogen, of oxygen content when you start functionalizing them because of the PEG, and when you do the fluorescing and the folic acid and the functionalities, you have essentially a further increase of nitrogen, which tell you essentially that the click chemistry probably had worked quite well. So in fact, analyzing the high resolution peak of the carbon, we see that we have the presence of the COH, the carboxyl function, after the first reaction one, essentially, and then the presence of the silicon after the reaction two, in order to prepare the carbon nanonions to be able to receive the fluorescein and the other and the folic acid. When we go to the TOF seams, we realize that try to identify the peaks, or the difference in peaks between these three different samples were absolutely almost impossible. But the multivariate analysis that is normally used in SIMS analysis of data came in our help, and so we were able to separate basically the pristine sample from the two other samples, functionalized in one case, and using the loading plot, we see that the negative score are essentially related mostly on the carbon hydrogen bonds, where the other are related to carbon oxygen and hydrogen bonds, and some other peaks are related also to silicon. But this is even more clear when we plot the principal component 1 versus principal component 2, and we look at the loading plots. We see that the carbon nanoions pristine are essentially related to the CXHY type of ions, and the other one are related to the carbon oxygen bond, and the third one is containing the silicon and silicon oxide kind of fragments. Okay, so also the XPS is showing even more increase in nitrogen when you functionalize the carbon nanonions with the fluorescein, the CNO5 here, and with the folic acid. And you see that you have a systematic increase in nitrogen content, as I show you, and there is no much shift. So there is no much free nitrogen. So this nitrogen is basically related to NNN or CN bond. So the SIMS also was able to pick up a lot of fragments, in this case in negative polarity, related to the fluorescein break of this kind of fragment, and also the fragment related to the click chemistry when we did the fluorescein attachment by the click chemistry. And also here we have these large fragments here, which correspond to mostly the molecular parent material, the fluorescein. And also attaching the folic acid after the fluorescein, you have essentially again the click chemistry here, and then you have the folic acid fragments that are appearing, and still the fluorescein fragments there. So in summary, for this part, I have to say that we demonstrate that although you can have different techniques that can tell you that your surface functionalization has worked indirectly, XPS and TOF seams give the possibility to essentially really identify the chemistry and the single molecules and the single fragments related to the molecules we attach to the surface. So we did also some cell viability study in order to see if this kind of material with this kind of functionalities were essentially toxic or not. And in fact, they are not toxic up to 20 microgram per liter, microgram per milliliter. At 20 microgram per milliliter, you start having essentially a decrease of the viability to about 70%. The other problem was to see if these nanoparticles were essentially internalized by cells. So we did with the ELA cells, and we can realize that they are definitely internalized, and they are internalized in the perinuclear region, but not in the nucleus. Also, we did some light and electron microscopy correlative CLEM analysis by looking at the confocal microscope in one side and looking at the same cells in by the TM, and reconstruct with the correlation. And as you can see here, we see that this kind of nanoparticle are essentially internalized, as we saw also with the confocal microscopy. And by sectioning and doing the TM, we see the agglomeration of the carbon nanoions into the lysosome. So these are quite interesting type of analysis that can be used to correlate a light microscopy with the TEM. And also, these demonstrate that the possibility to use the carbon nanoions as a marker for TEM analysis, which is not so easy to find marker for TEM analysis in cell. Here is another thing we did. Yeah. Targeting.
Sorry.
And non-targeting.
Yes, you have to be online.
Okay, I'm finished. So basically, the conclusion is essentially that the two cell line tested were essentially good for cytotoxicity, and essentially the CLEM analysis demonstrated that this kind of quite complex functionalized nanoparticle were able to be internalized by cells. And with that, I thank you for your attention, and I will be pleased to answer questions. Thank you.
Let's see. Can you hear me now?
Yes.
Okay, thank you so much for your nice presentation. Let's see if there is any question. And I can check from the chat. There is no question. Can I ask you a question?
Please.
Because I want to ask you how we can control the if they are uniform, the nanoparticles uniformly functionalize with this method.
With which method? I mean, okay, the dye you can yeah.
To compare.
Yes.
One is better.
Okay. There are two ways. I mean, one way is essentially to make a calibration curve, essentially by changing the amount of let's say anchoring point in the nanoparticles, and then you essentially attach the functionalities until you have a fully you reach a plateau. And from this calibration curve, at the end, you can calibrate or compare eventually a known sample by looking at the XPS concentration of nitrogen or carbon peaks, and so you can place your sample within the calibration curve.
Okay. Thank you so much for your great work. So we can go to the next presenter, Just Lotters, okay, with title of Demonstrator Systems for Real-Time Inline Measurements of Medicine Mixtures. Okay, please. Can you hear me?
Yes, I can hear you. So now you can see the screen, my screen?
Not yet.
Okay.
Refresh it.
Do you see it?
No. Share screen, and then you have to click on the screen.
Yeah, I shared the screen. And it's on your entire screen. And then I cannot click on share.
No, you should click on the not share screen. You should just click on the screen.
Ah, yes. So now you can see it?
Yeah.
Great. Okay, for the good watchers in the audience, you can see I added the word towards a demonstrator system because we wanted to do all kinds of experiments, but due to the COVID time, we were very much restricted. So we are working towards a demonstrator system for real-time inline measurement of medicine mixtures. My name is Joost Lutters, and the team you can see below of all the people of Bronkhorst and the University of Twente who worked on this topic. So as an introduction to the problem, that in hospitals there are neonatal intensive care units, and they use multi-infusion systems for prematurely born babies. And these systems, as you can see in the picture, are very complex, because in this picture here, you can see 8 different infusion pumps, but there are systems from more than 10 different infusion pumps simultaneously connected to the neonate, and they will administer medicine mixtures of more than 10 different drugs. But there are many complicated problems here because of the complexity of the setup, and due to the really low flow rates, sometimes below 1 milliliter per hour, severe problems can occur, which could potentially lead to injuries at the babies or even death. There were several investigations. For instance, the FDA, they received more than 56,000 reports of problems associated with infusion pumps, and also startup delays, flow rate variability has been seen. And at the University Hospital in Utrecht in the Netherlands, they saw that you think you administer some drugs in the mixture, but they saw that some individual drugs were either not administered to the patient or way too much present. So this could lead to these really dangerous situations. So at the moment, the flows and everything administered to the patient are not monitored. So we propose this solution as shown here. So here I hope you can see my cursor moving. So here you see the syringe pumps. In this case, it's a set of 3. And then we want to measure with Coriolis flow sensors the flow coming out of the tubes. And why Coriolis? Because that principle is independent of the physical properties of the liquids. So always the right flow rate is indicated. Then these 3 lines add here in the mixing block. And from the mixing block, the mixture is going through the multi-parameter chip, where we can really check are all the necessary drugs inside the mixture. Are they not too much? Are they not too less? And then it's provided to the patient. And then in case anything would go wrong, then this multi-parameter chip would give out some signals. This provides feedback information, so maybe a nurse can adjust the syringes manually, or maybe later on we can do it in a controlled way. So how does this measurement principle work? There is, here you see the multi-parameter sensor very schematically, And here we have a mixture of only 2 drugs, drug A and drug B. And then the mixture goes into the sensor system. And of course, it leaves the sensor system again towards the patient. And then we can measure flow, density, viscosity, maybe other parameters like permittivity. And this gives you the total parameter set of the mixture. And because we know which medicines we are in the mixture, via processing, we can decompose it into, for instance, in this case, 80% medicine A and 20% medicine B, because these drugs have different characteristics in different densities, different viscosities. And therefore, we can decompose every mixture into the individual drugs inside the mixture. This is more the mathematical model behind it. For instance, in this case, we have 4 different sensors. So like the flow sensor, the Coriolis flow sensor, the density sensor, the pressure sensor, and here's also a thermal sensor. And because we know the behavior of each sensor, we know which properties affect the output of each sensor. We can combine the values of these sensors, and then we can calculate, for instance, the viscosity. So how does that work, viscosity? That's with the Hagen-Poiseuille equation shown here. And therefore you need flow and density, and that you will get from the Coriolis sensor. On the other hand, other side, for instance, a thermal sensor can give you information about the heat capacity of the mixture, and combined with the flow rate and the density, it gives you the heat capacity. And if you input all these data in the system, then you can, for instance, determine the composition by solving the 4 equations with 4 unknowns. So in case you have 4 different drugs inside the mixture, you can tell from all these drugs the composition. And the other way could be via correlation. Maybe glucose has a good correlation with viscosity, so then you could relate the viscosity value directly to the glucose level. Or you could use combinations of these methods, or maybe even other methods. Because you want to connect a very tiny device to the patient, the final goal would be to have a MEMS chip, a MEMS multi-parameter measurement system. And here you see the chip. So the size is 7.5 millimeters wide and 15 millimeters long. This is the schematic, and here you see all the sensors, Coriolis, and pressure. And then you see how we use all this information to end up with the viscosity in this case. And then on the right side, you see the chip. This is the chip. It's fabricated and it's connected on a PCB with electronic circuitry, so we can have a readout of all the signals. So this is work in progress. The chip is there, as you can see. We already connected it, and we are building the total system. But of course, in the meantime, we want to demonstrate the feasibility of the concept. So we started with regular instruments. And that's shown here. So basically, it's the same concept. So here we have the pump in the picture, and the pump goes to the first pressure sensor. Then we have a capillary to have a pressure drop. Then we have the second pressure sensor. So here we can measure the differential pressure from the Hagen-Poiseuille equation. And finally, the flow goes to the Coriolis flow meter, which measures the mass flow and density, which are the other factors in the Hagen-Poiseuille equation. So with this setup, we can measure the viscosity of the mixture entering this system. And here you can see the real life setup with here the Coriolis flow and density meter. And here you can see the 2 pressure sensors, and in between you can see the capillary tube. And of course, we have to see which medication is most used in the neonatal intensive care units. And this information comes from the University Hospital in Utrecht in the Netherlands. We're also involved in this project. So apparently, many babies have problems with their blood pressure, and most of the time it's below the regular value, so hypotension. So they need dopamine or maybe dobutamine to regulate the blood pressure. And of course, also morphine is used as a painkiller, or maybe sedating like dormicum, Nucuron, and propofol. So these should all be administered. And also added sometimes, or most of the times, is a saline solution and glucose as a nutrition. And we chose to start with glucose, saline solution, dopamine, dobutamine as the pure single liquids, and then mixtures of those drugs. So here again, you see the measurement setup. Now, not only with the instruments in the setup, but also with the pumps. And here the pump, and here you see all the liquids under investigation. And then we started with glucose, and we administered glucose via the pump in different concentrations. So these concentrations ranged from 0.1% in water up to 5% in water. And what you can see here in this graph, on the y-axis you see the viscosity, on the x-axis you see the time, and you see water is around here, so the value of 1.03, something like that. And then you see there is a certain detection limit, so at low concentrations of glucose, 0.1 and 0.2, it's hardly distinguishable from water, but from higher concentrations, 0.5% and higher, it's clearly visible. You can distinguish the amount of glucose from water by determining the viscosity. Okay, so this is one way, this is the characterization. And the other way around, when you know this relation, for instance, in case you measure a value of 1.1 millipascal seconds, you can go in the left graph, you can go back from this value, and then you know, oh, this is the point, then you know that the glucose concentration apparently is 2.5 weight percent. So that's the measurement principle, how it will work. Of course, this is now only glucose. The other thing that we measured, because it was there, was paracetamol. We also measured that, and this has a viscosity change of circa 3% over the measurement range. So this is also nicely measurable with viscosity. And so we did more tests, and here they are summarized in a table. So water, EPA, ethanol. Of course, EPA and ethanol are not medication, but they were there. And then it's nice that you can check your system if it really works nicely, because of all these values, all these liquids, you have also theoretical values available. So that's nice to tune your system, and then later on you can do measurements with saline and glucose, and of course with other liquids. So we calibrated the system at a flow rate of 5 milliliters per hour for water, and in the lab it was relatively cold, 18 degrees. And I will show you in the next graph that viscosity is really temperature dependent. So it's essential that you know and monitor and control the temperature. So of course, therefore, this value, the deviation was 0% because we set it to that value. We calibrated the instrument, and then we just applied the other liquids as is through the system. And the nice thing is what you see is also at other flow rates, so 1 milliliter per hour, and with EPA and with ethanol. So the measured values of the viscosity were corresponding quite well to the theoretical values. So that's nice. And also for glucose, we found this value, and for saline solution. And what we see is that saline apparently cannot be distinguished from water via viscosity, because these values are too close to each other. So you cannot tell how much the saline concentration is in water. But for instance, glucose, you can clearly see at least above this 0.5% values of concentration. The difference for the different flow rates, they are caused by the inside temperature in the flow measure, in the flow meter, because at higher flow rates, the temperature inside the instrument is a little bit cooler, and here it's a little bit hotter, and that causes the differences in the measured values. So this is the graph of the viscosity versus the temperature, and the middle graph is for water, and here you see normally at 20 degrees centigrade it's 1 millipascal seconds, but you really see a strong change for other temperatures. So it's essential that you at least monitor the temperature so you can compensate for it, or you have a really well-conditioned room temperature. And I talked to the people from University Hospital Utrecht this morning, and they said maybe in some rooms, we think maybe in hospitals we have well-conditioned rooms, but it could be really terrible. So I guess the temperature sensor is essential. Then something else, we also did some density measurements, and that we did with the two blood pressure regulators, dobutamine and dopamine, and we used the ampules. So we did not put it into solution, but directly took it from the ampule. And then you see in this graph on the x-axis, it is the mass concentration of dopamine in dobutamine. So we start here with 0% dopamine in 1.25% of dobutamine, and then going to the right side, we go to the higher level of dopamine, 4%, and 0% of dobutamine. Oh, sorry. And what we see is a good variation with the density. On the y-axis, we see the density. So of water, it will be around 1,000 kilogram per cubic meter. And we see an increase of the density with the dopamine in dobutamine solution. So this is the characterization, and here the red lines again indicate how we could use the system in practice. So if you find this value of, for instance, circa 1007 kilograms per cubic meter, you go back into the curve, and then you see, okay, this has to be the concentration of 2% dopamine and 0.63% dobutamine. So summarizing all of these results, because these mainly were the single fluids. So from now on, we will have to characterize also the mixtures of all these liquids. But we think we can achieve these specifications. So for flow, it will be, so the conventional system, it's the 1 to 2 milliliters per minute and 0.2% reading accuracy. The MEMS, of course, will be a little bit lower. Pressure in the range of 1 to 5 bars, density, as mentioned here. Temperature between 15 and 30 degrees centigrade, accuracy of 0.2 degrees centigrade, and viscosity in the range of 0.5 to 5 millipascal seconds with an accuracy of 2%. So in summary and an outlook to the next stage of the research, drug delivery via multi-infusion that may cause severe adverse effects to patients at intensive care units and especially to neonates. At this moment, the flow and composition of the medicine mixture administered to the patients are not monitored. We developed a multi-parameter system for real-time and inline measurement of the medicine mixtures. And the chip version, which should be the envisioned final version, that's still a work in progress. So in order to prove the principle, we composed a conventional system of standard instruments, and that's being characterized. So for now, we measured the viscosities of the single fluids, water, paracetamol, glucose, and saline solution. And they can be distinguished from each other via viscosity, so except for saline. And for the medication, we already tested, that's dobutamine and dopamine. For the blood pressure regulation, we measure the density, and they can also be distinguished from each other. So next steps will be the characterization of mixtures, so binary and ternary, of saline, dobutamine, and dopamine, because apparently they are the most occurring drugs, to see the composition of these mixtures, of whether we can determine them from viscosity and density. So within the this is a European project. It's called Metrology for Drug Delivery Part 2. The website is drugmetrology.com. Here you see all the partners involved. And thank you for your attention.
Thank you so much for your nice presentation. Let's see if there is any question. And from the chat also there is no question. So I think we can close the section, and we will see tomorrow morning for the second day of the workshop. And for the people who are present here, I just want to remind you social dinner that we will see in Campano restaurants at 19:45. So thank you for your attention and have a good night. Goodbye.